So following on from gene flow, something we're going to pay a little bit of a closer attention to is this idea of random genetic drift. So random genetic drift itself is a change in the allele frequency of a population, not due to migration or anything like that, but simply due to chance events. So as this entire process relies on chance, it is completely random. It's also non-directional. And effectively, what this means is that the change in allele frequencies are going to be noticeable when there's a small number of breeding individuals or the population is quite small, or when gene flow is not occurring. So another example that we looked at in the previous lesson was, again, say the Vedoma people in Zimbabwe. So a very particular example of random genetic drift is the founder effect, and this is something that we're going to look at in a little bit of detail now. Now I've got a little bit more of a summary here. Feel free to pause the video, otherwise I'm going to scroll on now. So what is the founder effect? The founder effect is where a small subgroup of an original population colonize a new area. So if we have a look over on the right, we have this entire parent population, right? This large group of individuals. And within this large group of individuals, we have a particular frequency, doesn't really matter what it is right now, of red and purple alleles. Now, as the population migrates following that red arrow, you'll notice that we've taken a higher proportion of red to purple than was found in the original population. So this becomes the founder population, or the new population. And what you'll notice is that subgroup, that founder subgroup, is not genetically representative of the original population, where there was roughly, let's say, a 50-50 proportion between the red and the purple allele. Now, over a couple of generations, we see progressively, as we move down through the generations of that founder population, you can see that the allele frequency changes significantly, and then we have slightly more purple to red. And then eventually, after a few more generations, funnily enough, the red allele, whatever that is made to represent, is completely extinct from the population. And so these two populations, being the parent and the founder population, are no longer very common at all in terms of their genetic compatibility, or not genetic compatibility, their genetic representation with one another. So if we have a look, there are often, is the case with the founder effect, there can be barriers to gene flow. So these could be geographic or sociocultural, so things that we looked at in our prior lesson on gene flow. And this often results in no gene flow, right? So if there's a big mountain range dividing the parent population and the founder population, there's not going to be easy gene flow between these two groups. Same thing in the case of humans. Say there's a particular religious belief that's formed in the new group, or maybe a particular sociocultural belief within that group. Maybe they won't have any gene flow or any migration between those two groups. And what we see, again, over time, the key part here, is that the allele frequency changes significantly over time between these two populations. So a particular example, real-life example of this, is the Dunkers. Now, the Dunkers were part of a religious belief group, and they originally came from Germany, okay? And a very small group from Germany, made up of only about 50 families or so, moved to Pennsylvania in the United States. And their religion specifically prevented marrying from outside of their group. And so what we've got now is we had a large original population, a large original gene pool, and we have a new founded group with only 50 families, which is actually quite a small population. And what that led to is then inbreeding due to their religious beliefs, and this has resulted in significantly altered allele frequencies when we compare the US Dunker group to the modern day German Dunker group. So this is a representation here just showing how allele frequencies can change, especially when we introduce things like geographical barriers, right? So this mountain range that we have in the middle that I'm highlighting in yellow here. And then within these two populations, say, let's call this one the original population up the top here, we can have the immigration and emigration within that particular species, but maybe in this next group, or this, let's call this a founded group down here, we're getting very limited gene flow, if anything, between these two species. And what we're seeing down the bottom is we're getting the effect of this random genetic drift within members of the population.